Hello, everybody. Thank you again for joining. Again, I'll say it one more time. This session is being recorded. We will share it after the fact, once we get everything, everything set up and put onto YouTube. Thanks again for joining. And if you do want to ask any questions, which we do encourage, please use the Q&A box. You can see it down at the bottom of your screen there. Use that to ask any questions you may have. I'll do my best to moderate all of them and combine them when necessary, and have a great discussion with our two wonderful presenters today. So before we begin, if you don't already know, my name is Andrew Byrne. I'm the Marketing and Communications Director at BioNB out of Fredericton, New Brunswick. And I'd like to thank the other organizers who keep helping us create these episodes month after month, and that's the Nova Scotia Innovation Hub, CCNB Innov, and Ignite Atlantic. So thank you very much for continuing to help us and help the bioeconomy and the bioscience sectors grow. And so I will do a brief introduction of our speakers, and then we can let them talk about their phenomenal research. So first up today will be Dr. Stephanie MacQuarrie. She's an Associate Professor of Organic Chemistry at Cape Breton University and Associate Dean of Science and Technology. She obtained her BSc from Mount Allison University in 1996 and her PhD from Virginia Polytechnic Institute and State University in Organic Catalysis in 2005. In 2005, she joined Dr. Crudden's group at Queen's University, where she worked in the area of materials chemistry for 5 years. Dr. MacQuarrie's team focuses on finding high valuable applications for waste products, reducing the total carbon footprint of advanced materials, and the development of functional materials for electronics, catalysis, and absorption from biochars generated from various waste streams. Her program is funded through NSERC-DG, as well as many industry partnerships. She has over 45 peer-reviewed publications, has supervised over 50 HQP, including PDFs and graduate students via her adjunct status at MUN. And second presenting today will be Dr. Audrey Moores, who is an Associate Professor of Chemistry and Tier 2 Canada Research Chair in Green Chemistry from 2007 to 2017 at McGill University, where she started her independent career in 2007. She completed her PhD from the École Polytechnique, France in 2005 and received the Best Thesis Award that year. She was a postdoctoral fellow at Yale University in 2006, funded by a Lavoisier Fellowship from the European Union. She has served as co-associate director of the Center of Green Chemistry and Catalysis, has been the scientific director in the board of Green Centre Canada, and is the Associate Director of the Facility for Electron Microscopy Research, FEMR, at McGill. In 2019, she received a Fessenden Professorship Award by the Faculty of Science at McGill University towards the commercial development of her discovery on the clean transformation of crustacean shells into valuable biopolymer chitosan. So I'll let again, the presenters do their thing and inform you much better than I did what they're doing. And take it away. First up is Dr. Stephanie MacQuarrie.
Great. Thank you very much, Andrew. Thanks, everyone, for joining us today. I'm going to turn my video off and share my screen here. So I'm really pleased. Hopefully everybody can see my screen. If not, if someone can just unmute themselves and let me know, that would be great. First of all, I want to thank Atlantic BIOCON for inviting me to speak today and hosting this virtual speaker series, and also thank all of you for attending, and thank Dr. Morris for sharing this time slot with me. I'm excited to have the opportunity to tell you a little bit about a project that has been ongoing in my lab at Cape Breton University over the last 5 years, where we've been looking at the generation of crab charcoal and higher value products generated from that charcoal derived from crab body waste. So a little bit of background. About 5 years ago, Louisburg Seafoods, which is one of 6 crabbing industries off of Cape Breton Island, contacted our research group at CBU and asked us if we might be able to work with them to find a less costly way to deal with their waste issue. The crabbing process for generating the sort of the desired edible parts of the crab, speaking of snow crab here, is removal of the shoulders and leg for sale. And then that generates about 60% of the rest of the snow crab's body mass as what is considered a waste product. And so Louisburg Seafoods and the other crabbing companies on Cape Breton Island are responsible for getting rid of their waste during these crabbing seasons. And currently, in this year alone, for example, and it was kind of an odd year because of COVID, but even so, Louisburg Seafoods had to haul more than 150,000 tons of crab waste off of Cape Breton Island to landfill and compost facilities on the mainland of Nova Scotia. And so this is, you know, a serious issue for them because it is costly and requires them to pay for hauling fees, but also pay for landfilling and compost facility fees. Also, it's a stinky issue. So it creates environmental concerns for neighborhoods that are close to these landfill locations or compost facilities, because you can imagine that a product that looks like this probably doesn't smell that great either. And that is the case. So they were interested in working with us to see if we could find some way that we could get rid of this waste or use this waste to generate some kind of valuable product for them. Now, our group at CBU's expertise lies in the area of pyrolysis, which is essentially combustion of biomass, so something that maybe is considered a waste stream like the crab waste, under an inert atmosphere, so oxygen-free or oxygen-limited atmosphere, to generate products like crab biochar, for example. And the idea was that we would help the company reduce their carbon footprint by trying to find valuable products from what would normally be considered a waste stream for them, and hopefully provide some sort of income for them as well. So today I'm going to talk mostly about the applications that we found for one of the products produced during the pyrolysis process, crab char. We're going to look at some applications in remediation as a material product in things like concrete production, and as a soil conditioner for the agricultural groups. So what happens when we get the crab waste? The crab waste is this, we usually get it frozen. It has a very high water concentration, and it's extremely heterogeneous. So it's a combination of crab body parts, crab bodies, sometimes there's seaweed in there, and we don't do anything to purify that product. We just take it as it is. And so the biggest hurdle that we have when we get this crab body waste is the drying process. In order to get to a stabilized product that we can store for significant amounts of time in the lab, we have to dry it. And so this is truly the bottleneck of the whole project, because the drying process is expensive. We are really interested in trying to come up with unique ways that we can dry the crab that may be less energy intensive, but currently we use electric heat just as a convenient way to remove the water. So the total conversion of the crab, from the frozen crab to dried crab, is about 32% by weight. So we lose about 65% water mass when we dry the crab. But once we do dry the crab and grind it, we get this homogeneous material that you can see in this second image here in the baggie. It is a combination of a variety of whatever was in the crab waste that we got to begin with, but dried, but it's a much more homogeneous mixture. So it's largely calcium carbonate and chitin and other organics that would be found in the crab waste. But we have found that it's stable in airtight containers just at room temperature on the bench in the lab for up to 3 years. So it's fairly stabilizing as long as we're able to remove all the water from the crab waste when we receive it and dry it. Then we use this crab body powder as our starting material for the pyrolysis process. And so I'm not going to get into too much detail around the process of pyrolysis. There's 2 major types of pyrolysis. There's slow and fast. In our lab, our focus is largely on slow pyrolysis, but we have the ability to do both. And what pyrolysis is basically you take your biomass feedstock that's dried, feed it into a burner. The burner can be powered with electric heat, or it could be through, utilizing the combustible gases that are produced and burned to provide the heat. And the larger material components or larger compounds or particles are collected in some sort of collection matter, whether it's a cyclone or a filtration unit, as what is considered the biochar, the char collection part of the pyrolysis process. The smaller molecules will either be condensed to form the bio-oil and water phase and collected as bio-oil. And then the very small molecules and the combustible gases can be recycled in the system to be burned, or they could be off-gassed into the atmosphere, which is not a great way to do pyrolysis, or they can be collected in a gas collection unit. And so to keep the whole entire process fairly carbon neutral, it's ideal that you can use all the products that are produced from the pyrolysis process. You want to collect the char, collect the oil, and somehow collect and use the gases as well. Our main focus has been on the two major products of pyrolysis, which is the crab biochar and applications around this, as well as we've done a little bit of work on the crab bio-oil that's produced. So as I mentioned, we're able to run both fast and slow pyrolysis. The difference is that a slow pyrolysis unit generally heats at lower temperatures with slower temperature ramps for longer periods of time, where a faster pyrolysis unit will heat to higher temperatures with faster ramps for shorter periods of time, both under inert atmospheres. What you can see is that the yield of the char is generally the same, no matter which conditions we use. So we're getting about 50 to 55% crab biochar through pyrolysis. But depending on if we use fast or slow pyrolysis, we'll get either a higher bio-oil yield or a higher water yield. So fast pyrolysis gives us higher bio-oil yields, and slow will give us higher water yields. As I said, we haven't done a whole lot of work with the crab bio-oil that's produced so far, but what we have started looking at is whether it could possibly be used as a replacement biofuel. So most of the work done in the area of biofuel has focused on using woody biomass as the starting biomass to produce the bio-oil and using fast pyrolysis. And woody biomass, especially woody biomass from softwoods, generates an oil product that has a higher heating value. So it's basically the gross energy of the oil of about 17 megajoules per kilogram. Now, compare that to what is currently used as bunker fuel. So most bunker fuels from fossil fuels will have higher heating values of around 40 megajoules per kilogram. The crab bio-oil is closer to bunker fuel, around 30 megajoules per kilogram. And this is really promising, and we are excited by this. We think it's probably because it has a fairly low water content and a high pH compared to forestry residue bio-oils that are typically acidic and have a much higher water content. We are now working on identifying the chemical composition of the crab bio-oil and looking into that more thoroughly. But as I mentioned, our main focus has been the crab biochar because it's the most high yielding product of the pyrolysis. And we wanted to look at, getting a really good handle on what the crab biochar was composed of. So we've done a really thorough characterization of this material. I'm just going to show you a little bit of our evidence that tells us what the crab biochar is composed of today. Notably, the biochar itself is also very basic. It has a pH of around 10. And that makes sense when we look at the composition of the biochar. So the bottom spectrum is an FTIR spectrum, and the peaks that are shown in light green, on my screen anyways, are pure calcium carbonate. And when you compare that to the peaks that are shown in the sort of a yellowish color, that is the 100% crab biochar, and you can see that it is largely matching with the calcium carbonate. But we also have this peak associated with calcium oxide, which is not terribly surprising because the pyrolysis, although it's a controlled temperature setting, you do get areas of very hot temperatures that go above your 450 degrees Celsius. And so that will facilitate the conversion of calcium carbonate to calcium oxide, but the concentration is fairly low. the crab biochar has an okay surface area of about 30 meters, between 15 and 30 meters squared per gram, and some limited porosity. So this is a SEM image of the crab biochar in an area that we found to be quite porous. This is most likely due to the organic components of the crab waste that is left within the shell. But because of this porosity and this reasonable surface area and the basic nature of the material, we thought it might be useful to look at this crab biochar as a possible remediation material. So being in Cape Breton, the coal mining industry has been prevalent over the last 100 years, and remediation of coal mines is still something that is quite important in this area. So we decided to look at whether this basic porous crab-based biochar could be used in acid mine water neutralization. So we had some acid mine water that had a starting pH of about 3, so fairly acidic. And we looked at dosing in a batch. So this is a batch experiment where we would literally just put in, a dose of the crab biochar in 100 mils of our acidic mine water solution. And we could see over a 2-hour time frame that we were able to get neutralization even at very low doses of the crab biochar. And so as we increase our concentration of crab biochar and we go up this graph, we start to get, neutral pH at around 7. But then we start to see a more basic pH all the way up to about 9. This isn't that surprising because the calcium carbonate that is, the main composition of the crab biochar is soluble. So it starts to leach into solution and increase the pH of the overall mine water to about 9. It stops about 9. So it's promising, but we're still working on whether or not this is something that is usable in the field. We also wanted to look at whether the crab biochar would be able to remove heavy metals from the acid mine water as well. We looked at a variety of heavy metals. What I'm showing here is the removal of iron from the acidic mine water. And so the starting acid mine water had a concentration of iron of about 7,000 parts per billion. And so after the maximum dose of 0.5 grams per 100 mils over a 1-hour period, we see very, very little iron left in solution. So it's almost unmeasurable. And we could even see iron absorption just using crab powder, so non-pyralized crab powder. So those shells just dried and ground up were able to absorb the iron as well from solution. So this is some of the work we've done looking at the crab biochar for remediation. We've also started collaborating just over the last 1.5 years with cement chemists and cement scientists who have helped us look at whether or not the crab biochar, the crab powder itself, could be used in the cement making process. So part of concrete composition, as you can see in this image here, is about 10 to 11% cement. The rest is, gravel, crushed stone, sand, and water. And the cement that is used in concrete is generated using limestone from lime quarries. And that liming process is really, limestone mining process is really energy intensive and actually makes up for about 10% of global man-made CO2 emissions. And so the cement is made by heating the limestone that is mined in the quarry at about 1500 degrees Celsius with other components of the cement in a kiln, and that generates the calcium oxide that is required for the chemical composition of cement. So the main issue is that a lot of concrete industries and cement industries are located near or within a limestone quarry because they rely so heavily on this limestone in order for their production of the materials. And aside from the fact that the mining process can be environmentally harsh, they also want to extend their lifetime of the industry and their location as long as possible. And so concrete and cement industries are interested in looking at additives that could be used as sort of replacing some concentration of the limestone that they're currently mining in the cement making process. Now, we have only looked at the addition of crab char to the concrete itself. We were just starting to send some of the crab body powder to a local cement industry for production within the kiln before COVID-19 sort of shut all that down. But we wanted to look at whether the addition of crab char itself to the formation of a concrete pillar, just something we could make in the lab, would have any kind of impact on the overall structure of the concrete before we went further within this project. And what we found that was, with the addition of only 5% crab char concrete compared to the addition of nothing, just making the concrete using the regular recipe, we saw increases in compressive, tensile, and flexural strength. So that was a positive result. There was no negative impact anyways on the addition of the crab char. I would say this is probably the highest value application we've looked at so far, as industrial limestone production in 2017, which is the sort of latest statistic I could find, was about 1.3 billion metric tons, which was valued at about $25 billion. So it's a really high-end application, high-value application for potential for this crab powder or crab biochar. However, there's competition for mined limestone. It doesn't all go into the cement industry. In fact, high volumes of the limed limestone are sold at lower value to the agriculture industry for soil conditioner. So because soil acidification is a major issue in the agricultural sector, and it's a natural process, and it's a result of increased or accelerated crop production. So as you know, the number of humans that live on Earth increases, the more food we have to make, the more quickly we're trying to grow food, the more fertilizers we're using. What ends up happening is we acidify our fields and our crop and our land, and that causes diminished crop production. So limestone or lime is often used as a soil conditioner to help reduce the pH of the soil. Now, I'm a chemist, so we collaborate with some of our plant biologist specialists here at Cape Breton University in the Verschuren Center to do these planting trials. And we have the data to back these images up, but I wanted to share this little experiment with everyone as well. So what we did was we took a model plant that would be really highly impacted by acidic soil. So we chose barley, and we did a trial of over 40 different plants. And we looked at the barley plant growth with no conditioner to an acidic soil, with the addition of lime from, that you could just buy at Home Depot, to the acidic soil, and the addition of crab char to the acidic soil. And we looked at a variety of different metrics from the plants over this experiment, but I'm just showing you sort of something that you can visualize. What you can see is that the barley that had no conditioner only sprouted a few barley plants versus the cups that had both lime and crab char in them. They had much greater sprout rate. And then we also looked at the chlorophyll concentration, excuse me, the chlorophyll concentration and the shoot length and the sugar production in these plants as well, and found that in both cases, the addition of lime and the addition of crab char, we saw increased plant health. So we have a low sugar concentration, which would mean the plant was not under stress during its growth time. We've also looked at strawberry plants, and this past season we did a field trial with pumpkin plants, so that data is forthcoming as well. We actually think there's probably a lot of applications for the crab body powder itself, or the crab biochar that's produced through the pyrolysis process. In fact, many of the applications that lime is currently used in you could envision substituting, at least partially, crab body powder or crab biochar in those applications. And this page just shows a couple of those potential applications. So those are some of the applications that we've spent time investigating for the use of crab biochar. At Cape Breton University, most of the work is done by undergraduate students. This was my research group last summer when we could all get together and not have masks on. And they're almost entirely undergraduate students, with 1 or 2 postdocs and research assistants thrown in there. And so they do this work. They're excellent researchers. I'm really proud of the work that they do. They don't complain about the smell of the crab waste when they have to use it. And I would just want to say thank you to all of the students that did this work, and thank you to everyone who's listening today. And one more thank you to Atlantic BIOCON for the invitation. And I welcome any questions either now or at the end of Dr. Moore's talk.
Here we go. Thank you very much, Stephanie. That was fantastic. I particularly enjoyed the circular nature of the pyrolysis machine that you showed. I just wanted to let everybody know that we're going to do a question and answer period at the end because I feel like some of these questions you both may be able to play off each other. And so we'll go right into Audrey Moore's presentation now. So if you could please unmute yourself and turn on your video there, Audrey, and we'll get started. Perfect.
Yeah, absolutely. So let me get this organized. Sorry. Hi. So do you hear me okay?
Mm-hmm.
Wonderful. Thank you so much. So it really is a great pleasure to be here today, especially sharing the scene with Stephanie. I've been following her work, and I just really excited to be presenting this research with her. It's really interesting as well that we have so much in common. I'm going to talk a lot about crab as well, so this is really cool. So today I'm going to walk you through a little bit of the research we've done, especially trying to use crab in a different way and trying to solve some of the issues that we have in sustainability, in particular in the question of plastic. So as you know, we are surrounded with plastics, and plastic is really becoming a huge problem. It is impacting our lives in so many different ways. It's impacting ecosystems around us. We have microplastics that we can see in the ocean. It forms these big gyres that are akin to another continent in the middle of many of the oceans. We also have issues with the microplastics. So plastics that are either formed very small and will leak into the environment, or plastics that become very small by fragmentation. And there's been lots of work going on, such as here, this Ocean Cleanup Initiative, which is a large buoy here that has been tested offshore San Francisco, where they're trying to actually use that to collect plastics and then destroy it. So in Canada, this problem of plastic is particularly acute. We generate a lot of plastic, we use a lot of plastic, but only 9% of this plastic is effectively recycled. A lot of it is landfilled and completely lost, and a significant portion is actually leaking to the environment. And as we think about, especially the maritime provinces, this is particularly dear to our hearts to protect the ecosystem here. So this is with some of this reasoning that we started looking into this question. We have next to us materials that are very similar to plastics, that are plastics actually, and that we could use. So I don't know if, like me, you love lobster, but when I eat lobster, there's always this moment when you crack the shell of lobster and it's really hard and it's resistant, and you can also feel the flexibility of the material as you break it. So this is bound to make a really great material that we could use for making plastics. So it turns out that the material that makes this flexible and tough property of lobster is actually coming from a polymer called chitin, which is present inside the lobster, and that Stephanie mentioned a little bit in her presentation. This chitin polymer is present in crustacean shell, but also in insect shell, which is going to become relevant as well for my presentation. And if we think about that, there's about 6 to 8 million tons of waste crab, shrimp, and lobster shells that are produced every year globally. And this waste is currently very little used. I mean, as I'll show in a minute, we use it to make a little bit this material called chitosan, but it's only a tiny fraction of it that is used, and the rest is discarded. And as Stephanie mentioned, it's actually a big problem for the environment. So if you take a crustacean shell, we can dive into this a little bit to understand the structure a bit more. So again, following the past presentation, we have learned that inside the crustacean shell, we find this polymer called chitin. There's also some proteins that are left, which, if you can extract them, are interesting materials as nutraceutics. And through this network of two polymers, we have calcium carbonate that are present as small particles, and they confer toughness to the ensemble. What you want to do when you take this is you want to find a way to take out a material that is interesting in it, which is chitin. So what you'll do is you'll extract chitin from the rest, and that calls for the use of a lot of chemistry. So you usually will expose them to corrosive materials and heat. Then from chitin, it's a good material, but you can actually turn them into chitosan, which is an even better material as it has a lot of cool applications in sutures, water filters, or antibacterial materials, and so on and so forth. But the issue with this is that it really calls for very dirty chemistry. This process here necessitates to work with concentrated solution of sodium hydroxide, which means that you have a very viscous, corrosive liquid that you need to heat. You need to add this liquid in large excess to the chitin that you're treating. And this is making for very, first, unsafe workplace, but also it's generating a lot of downstream water, industrial waters that are difficult to dispose of. They're very basic, and it's not something cool. So I know that in Quebec, for instance, they used to have a plant that was doing this transformation in Gaspé, and they actually closed it a few years back because it was just too complicated to handle this, the difficulty associated with especially the waste stream of this plant. So in my lab, we work with a technology called mechanochemistry. So this is the use of devices that are going to mix stuff, but without the use of solvent. So in these kinds of equipment, we're mixing powders together. And what we discover doing this is that a lot of chemistry happens, even if you don't have any solvent around it. So you can use very simple stuff like mortar and pestle, and for a number of things we do, it actually works very well, but we also use more rigorous methods using planetary mills or vibrational mills, or even extrusion, which is a flow version of this mechanochemistry. Another thing that we do is we use aging. So not that kind of aging, right? But aging whereby we actually leave powders together to react, and we just wait. So sometimes we will expose them to a different environment, and we find that a lot of chemistry happens as we just let chemicals sit together. So what we did is we developed a method whereby combining mechanochemistry and aging with humidity, we are able to perform the same chemistry I mentioned earlier, but this time without the corrosive liquid and difficult to work environment. So I'm starting here with chitin, which is crystalline, and I'm going to treat it by mechanochemistry. These bowls here are here to indicate we're going to do mechanochemistry. And it's going to make my chitin more amorphous, so it's easier to play with. And then I will mix it very shortly with NaOH. And at the end of the day, I let it age for some days under high humidity, and it's going to fully convert my chitin into chitosan, which is actually pretty cool because it will use far less energy than the process before. It uses about 1 order of magnitude less water, and it uses, again, about 1 order of magnitude less chemicals to do this. So it's really much leaner in terms of green chemistry. Something else that we realize as we do this is that we also make chitosans that have much longer polymeric chains. So we're preserving the structure of the initial chitin much better, and we've made chitosans that have completely new properties. And in particular, this chitosan does not dissolve in water or acidic solution, which means that we can start to make stuff with it. We can make a cup, we can make a piece of something that's not going to dissolve in water. So here we did this from chitin, but we also showed that we could use even powder from crustacean shell directly, or even insects. And we get very good results in terms of deacetylation, which is the marker that will tell us that we have made chitosan. We won't have something in the 90s, and as you see here, most of them give that. And as high intrinsic viscosity here is an indication of how long the polymer is, and these means that we have really, really long polymers. Again, something that is unusual to find for this reaction. What's cool is that insects work as well. So fly larvae and beetle give very good results, which is interesting because as we're moving along, we also know that people are gonna look more and more into using insects for protein to feed humans. And this is some, you know, the chitin from insect waste is also going to be a growing waste that we can tap onto. So we've made this transformation from chitin to chitosan, and now are developing methods to mold this into different materials. And that's really something that we're looking forward to developing even more. We're also looking into going more from, you know, collecting more of the calcium carbonate from the shell as we do this transformation. So there's a lot of different developments that's happening in my group. One thing that we're particularly interested in is actually developing this for sources of crabs that are unusual, and in particular, we're partnered with Kejimkujik Park Seaside. I know some of you are down in Nova Scotia, so hi to you guys. We have partnered with them because they have Kejimkujik is a beautiful natural park down the coast in Nova Scotia, about an hour and a half down from Halifax, and this is a place where they have a lot of invasive crab, these green crabs that you can see here, and they generate a lot of waste from it. So they've asked us if we could take this and take the shell from these guys and turn them into our materials. So this is a project for which we did some fundraising via a crowdfunding platform and the Fathom Fund from MioPar. So we successfully got this project going. But now we're desperate to hire a postdoctoral fellow to do the research. So if you know someone interested, I really encourage you to spread this ad. We're really looking for someone to do this work now. Finally, I'll just wanna zoom out a little bit on other things that we're doing with the similar project. This mechanochemical method is really powerful. So we've developed mechanochemical transformations for polymers, for a specific polymer called MPEG, which is a polymer that is used for a lot of applications, from food additives to medical stuff to materials, and we can very easily functionalize it to make a lot of functional groups, again by mechanochemistry. We have also made a transformation to make polyketones, which are a popular thermoplastic polymer. We're making it more hydrophilic, which is difficult to do normally, and again, mechanochemistry can help do that. And finally, in a different line of thought, we're also working with chitin and chitosan and turning them to the nanoscale. And we make this little whisker-shaped materials. We are working in partnership with NRC, where they're making a very nice methodology for this transformation from bulk polymers to nanoscale polymers, and we're looking at applications in catalysis and functional materials. So with this, I would like to thank you very much for your attention. Thank the support, financial support from a bunch of people who are supporting us. Of course, my group, without whom I couldn't do any of that. And I really wanted to show this picture here to you today, because this is the last picture we took before we went all on confinement. This was taken on March 13th, and it was the last group meeting, and I have, you can see here where we're disinfecting our table before leaving for good, actually, before. So we haven't seen each other since, at least not in person. So I wanted to thank you very much for your attention, and if you have any questions, I look forward to answering them.
Perfect. Thank you very much, Audrey, for that great presentation. Thanks again to Stephanie as well. You both are doing wonderful research and necessary research, in my opinion. So Stephanie did answer two of the questions, but I think that the rest of the audience would like to hear the answers as well. So Stephanie, two things. Have you thought about testing neutralization at different biochar soil ratios? And what is the standard deviation of measurements?
Yeah, so we looked, I wasn't sure if I answered the question correctly anyway, so this gives me a chance to figure that out. So for the different concentrations for neutralization at different biochar soil ratios. For the plant studies, we looked at a whole variety of different concentrations of biochar, crab biochar, and I just didn't share a lot of that data today. But what we found was that there's definitely a limit. It's same with lime. Once you get to a too high of a concentration, the seeds don't germinate, and you don't get any plant growth at all. And so I'd be lying to you if I tried to tell you what the optimal concentration of crab char was for the barley growth at this very moment, but it was less than 10% of overall soil mass for addition of crab biochar. But what we did was we determined the exact concentration of calcium carbonate in the crab biochar and added the same concentration of lime. So we were doing a direct comparison of lime concentrations. We suspect that the addition of the charcoal component of the crab biochar is beneficial to the soil as well. But there's a lot of mixed literature out there on using charcoal in agriculture. So, and then for the second question, I believe the question was related to standard deviation for our measurements in terms of the yields. For the solid yields, we get for biomass conversion to biochar for the crab waste, we get usually within 5% of the same yield for biochar. The biggest variation we see is with the liquids, and that's most likely because of the fact that the starting biomass is super heterogeneous. So we may be starting with like a lot of organic biomass if it's a really meaty body, or it could be a lot more shell based and a little bit drier, and then we would have lower water concentration. Hopefully that answers the questions.
Excellent, thank you. I'm sure if it didn't, then they'll probably put it in the chat there and the Q&A box as well. But I do have some more questions, of course. These are very exciting topics. So do you have an idea of the scale of annual chitin production / waste generation? And how does this compare to annual plastics production?
So so thanks a lot, Hannah, for asking the question. And hi, Hannah, how are you doing? Nice to see you here. So as I mentioned, we estimate that there is between 6 and 8 million tons of crustaceans waste that's generating globally on an annual basis. That is actually in order of magnitude about the amount of plastics that is leaked to the environment globally as well. So if you compare those two numbers, they're actually not that different. Now, the question you might ask is it the relevant thing that you have to compare? So first of all, I'm mentioning the wasting crustacean shell, so you first need to look at the fact that only about a third of it is chitin, so the rest will be calcium carbonates and a little bit of protein. So calcium carbonates, if you could extract chitin and calcium carbonate nicely and separate them, you could actually envisage doing the nice chemistry of Stephanie with the calcium carbonate and then use the chitin to make higher, high value compound. Then I think also something else to see is that you really want to make sure when you look at plastic that you really make the plastics that you need for the purpose that you need. So if you want to inject new plastics in the market, my feeling is that it needs to be biodegradable plastics for usage where there's a high chance that it's going to get into the environment. So let's look at all the plastics that actually end up in the environment and figure out how we can back engineer those plastics so that they would degrade if they go in the environment. If you do that, you find that some of it comes from single-use plastics. So this is where you need to say, okay, we need to get rid of some single-use plastic, but maybe for other usage where we really need it, this is where a biodegradable plastics from chitin or from something else could be really cool. But it turns out that, and there's some really cool research done actually in the Maritimes about that, it turns out that a lot of the plastics you found in the ocean in particular actually come from fisheries. In fact, it's the #1 plastic you find in volume. And that's interesting because that's actually a very big engineering question. How do you engineer fish nets that would degrade, if ever, they're released to the environment? And that's so that's a really complicated question for itself. And then is chitin the best solution to that? I don't know, but I think what we have to do as chemists is try to provide toolbox to help as much as possible.
Excellent. That's great. Thank you for that. Great answer. And we have a question for both of you. So for Audrey and Stephanie, how much protein and oils in crab shell, snow crab and green crab, or the lobster shell?
So it's going to be different for all the different species. Snow crab that we work with, we just work with snow crab. We're interested in looking at green crab as well, but the reason that we are working with snow crab is because of the local industry. But the snow crabs have a harder shell than the green crabs, and they have I believe they have a higher calcium carbonate concentration. So I mean, it varies, but I think it's around 50% calcium carbonate, and then the rest would be chitin and proteins from the shells themselves. Probably more chitin than other proteins, but that'd be for snow crab specifically.
Perfect. And do you have anything to add there, Audrey? Or
yeah, I think that's the numbers that I have. So we didn't do ourselves any measurements outside of chitin. We know that we have from lobster shells and crabs between 30 and 40% of chitin, and the rest is calcium carbonate and proteins. And my understanding was that, yeah, protein would be 10 to 20%. So I think it's consistent.
Mm-hmm. Thanks a lot. And so something that I'm quite interested in as well is you both work with things in the ocean. You both work with animals in the ocean. And so if the ocean waters are warming with climate change, how can you expect this to affect your particular industry or research in general? And, would you expect to see more or less? Because it could go either way depending on what we're talking about these days. Could you expect to see more or less of the species you're currently working in as the temperatures rise?
Go ahead, Audrey.
Yeah, no, it's a good question. I think that my philosophy, in research is to try to figure out what we can do as chemists to answer the problems that are around us. So try to find something meaningful that we can do to improve. And we're also trying to advance knowledge in general. So what do we understand about these things? I mean, crab shells and crustacean shells in general are very interesting, high-performing materials. And they are studied, but there's still a lot of question about how they're made and how everything is entangled and how you can disentangle them and so on. So my position is that no matter what we do here, we're going to have more knowledge, we're going to understand them better, and say if tomorrow in Breton or in Kijim or whatever, it's a different species, suddenly we'll adapt to that. But with the knowledge that we've had, I think it's still going to be super useful. And I think that and some of what we do can be transferable. I mean, we did work a lot on cellulose, for instance, in my group for many tours, and was transferring a lot of that knowledge now to chitin and chitosan. So I think that there's lots of stuff that we can learn from different topics, and I don't think this is mutually exclusive in any way.
Yeah, I share the same opinion. I think that we sort of adapt to what the issue is. We're mostly interested in the fundamental aspects of our research, and we present much of that today, or at least I didn't. It was mostly applications. But I never ever thought I would be doing anything with snow crab until 5 years ago. So but I think what we're learning from that is that there is value in a lot of what is considered to be waste streams, and that we can look for ways. For example, a great example is that we spend quite a lot of our time trying to figure out how we can remove the water, maybe reuse the water in the crab bodies, and that would be a transferable skill that could be used for a variety of different waste streams, hopefully.
Fantastic. And have you ever compared the life cycle costs of using the crab lobster waste into an anaerobic digester to produce biogas instead of the energy to make new products?
Nope. No,
not there yet.
No.
No worries. And so, another question here from Benjamin. Both presentations were interesting, great science, and he's wondering if you could further discuss the next steps of your research into the translational aspects under more real-life scales, I guess.
Yeah, so I mean, in my case, we've been working with with a number of companies. I mean, what is my own difficulty right now is that we've had a lot of interest from different companies that are on the shell waste side of things, who are desperate to find something to do with their crustaceans waste. And we've also received some interest from downstream companies that are making plastic for one application or the other and are looking to see how they could blend this with what they're doing. What is complicated is to really bridge the gap between these two. And in particular, it's difficult for us right now to figure out what's the most realistic outcome of making this new chitosan. So are we going for biomedical applications? Are we going for bioplastics application, sutures, whatever? And based on this, it's very different research that we need to launch and different collaborators that we need to develop partnership with and so on. So I'm a little stuck right now, and COVID came at a really odd time for me because I was I was hiring for this at that moment, and it just stopped all the hires. And but now the labs are open. So again, if you're a postdoc interested in this, email me right away and we can chat, because I'm dying to restart this, and then have a realistic partnership with people as we move forward.
Fantastic.
Yeah, I think you know, one of the biggest hurdles, I guess, is for us is that we have spent a lot of time looking, like really getting a good idea in terms of the composition and characterization of the products that are generated through the process of pyrolysis for this biomass starting material, and then looking at applications. And the same with Audrey, we have a lot of interest from the fisheries industry to help them figure out how they can, utilize this waste or get rid of this waste and stop spending money on it. And then we can show applications of the products in these various fields that I talked about today, but there's a middle person missing who wants to take the waste and generate the value added products from them so that the applications could be used for like real life applications, for example. And I think that's the part that's sort of missing at this moment. And that's true for, the general field of pyrolysis across the world. It's sort of always on the brink of being something people are interested in. And hopefully in the next couple of years, we'll see it move forward at a greater rate.
Excellent. Hopefully so. And so one more question for each of you before I wrap up. So Stephanie, then what are some of your processing challenges? Again, with COVID, one of the big stops in the fisheries was the processing plants and getting workers and all this kind of stuff. So what are some of your processing challenges due to the seasonal nature of the snow crab fishery?
Yeah, so the biggest challenge that we faced this year was storage, because of the fact that we couldn't get in during the crabbing season and process the crab to get to the dry, stable ground state where we could, and not ground state, ground state, not like ground state in chemistry, but that we could store it in a bag and keep it there for, a couple of years until we could process it with the pyrolysis units. So we were really lucky because the seafood company that we worked with was able to store the waste for us in their walk-in freezers, and we just can go pick it up. But we're working in a university size lab. We only have a small freezer to store the waste, and you're storing 60% unusable water and only getting a small amount of product out of that. So that's the biggest issue that we have. And again, just drying it, like we're using an oven at the moment, and I know that's not the most energy smart way to do it, but we just haven't figured that out yet.
Yeah. Great, great answer. And for Audrey, similar question. Do you have a year-round supply of your crustaceans? Do you have seasonal challenges as well?
Yeah, so for the green crab project in particular, so they have also harvesting seasons, and similar to what Stephanie was saying, they're freezing it, and then we're getting the frozen ones, and this is how we work with that. We, for the crab shell, crab, lobster, and shrimp shells that we've been using, we were supplied with dried shells that have been where the meat was removed. So this is something that some companies that are doing transformation are doing. They're drying their shells. So that's a very nice material to work with. But as we move forward, it's true that we're going to need to, I mean, it's particularly the green crab project is one where we're going to need to work with live, full of water stuff. And maybe I'll call Stephanie when this happens.
Perfect.
For her help and advice. But yeah, from frozen stuff then is how we play that.
Okay. Thanks again to both of you for taking the time out of your busy schedules to talk to us, to talk to the BIOCON audience, and to allow us to kind of promote the phenomenal research that both of you are doing in this ecosystem. And just before we leave, I just wanted to give one more shout out to the folks that continue to help us here at BioND throw these BIOCON episodes off, right? The Nova Scotia Innovation Hub and CCMB Innov and Ignite Atlantic. So thank you to all those organizations as well. And one more thank you to both of you, and thank you to everybody for attending. So look forward to continuing the conversation. I used to say offline, but continue it online in a different format. So have a great day, everybody.
Great. Thank you, everyone, for attending. Thanks. See you later.
See ya.
See you later. Bye. Thanks,
Bye.
BioCon.